recently completed his PhD in bioinformatics, jointly carried out at the Technical University of Munich, the LMU, and the DZNE. He's the developer of AA analysis, a framework for explainable protein prediction, and in his talk he will show how explainable AI and novel algorithms uncover γ-Secretase substrate recognition.
Yes, yeah, thank you very much for the invitation to speak. It's actually a pleasure to see as well explainability here, having such a big space. So the title of the talk is Charting γ-Secretase Substrates by Explainable AI, and this is essentially a protein prediction problem. So to understand protein function prediction, I have to start with this fundamental paradigm of protein biology. So a protein sequence defines the structure of a protein, defines the function of a protein. The sequence of a protein is a chain of amino acids. There are different ones, for example, alanine, which is small and hydrophobic, and there's, for example, lysine, which is large and hydrophilic due to its positive charge. So the sequence you can see here is actually adopting a helical conformation, so-called alpha helix. And there are other important secondary structure elements, such as the beta sheet or random coils. And these together form the three-dimensional structure of proteins, exemplified here by γ-Secretase. Generally speaking, the function of a protein is a specific interaction of one protein with other biomolecules. And one very important example here is our proteases, such as γ-Secretase, which are molecular scissors. So it means that they can recognize other proteins and cleave them then into sequence parts. And the proteins which are cleaved are called substrates. And the aim of this project is to understand how γ-Secretase is recognizing its substrates and then to perform predictions. So why is this important? γ-Secretase is an intermembrane protease which is critically involved in Alzheimer's disease. So it's in the membrane, and the substrates are single-spanning type 1 transmembrane proteins, and they have one helical transmembrane domain, TMD, highlighted in green, and they have juxtamembrane domains, N and C terminally, highlighted in blue. And one very important substrate of γ-Secretase is the amyloid precursor protein, in short APP. So if we have a mutation APP on γ-Secretase, APP is miscleaved, leading to a 42 amino acid long cleavage product instead of a 40 amino acid long one. And these little bit longer forms are actually then aggregating, forming plaques, which ultimately lead to a neurodegeneration called Alzheimer's disease. So a long-standing question in the field of Alzheimer's disease is, how does γ-Secretase recognize its substrates? So here are 150 substrates known, such as APP or the cancer-related Notch1. However, there's no consensus motive or no characteristic properties. So in protein function prediction or to predict substrates of proteases, we always look for such amino acid motifs. So the starting point, the key question was, is there a common yet unknown property defining how γ-Secretase can discriminate between substrates and non-substrates? And to tackle this problem, I developed a new interpretable feature engineering algorithm called comparative physicochemical profiling, in short CPP. So CPP takes two sets of protein sequences and aims to identify the most distinctive features between these two sets. Here you can see our workflow. As explained, we use two protein sequence sets as an input and different amino acid scales. And then CPP creates in total 200,000 different features, and using statistical filtering to filter them down to the top 100 features, which then can be used for machine learning. So as a test data set, we used an expert-curated set of substrates containing 68 substrates called SUBEXPERT, and as a reference data set, we used 862 single-span type 1 transmembrane proteins, referred to as others, where we did not know whether they're substrates or non-substrates, so it's an unlabeled data set. And then as property scales, so they're properties like volume, polarity, or charge, and we organized them into an ontology called A ontology, comprising in total 600 of such scales, which we min-max normalized. And to select the redundancy reduced set of these scales, we developed another clustering-based algorithm called AA-clust. And now to perform machine learning, we of course not only need positive data, the substrates, but as well negative data, non-substrates. And there we just had 15 known non-substrates. So the data set is too small to perform proper predictions. And to tackle this problem, we developed a new deterministic positive unlabeled learning algorithm called DPO-Learn. And this aims to identify from the unlabeled data reliable negatives. in this way. So DPU Learn compresses the complete feature space in subspaces using principal component analysis, and then for each subspace we select the unlabeled data with the greatest distance to the positive one as negatives. And the number is actually depending on the amount of explained variance of the respective principal component. And this is a generic method which can be used for any positive unlabeled data problem. And it's important that it's deterministic. Because of this, we get 100% reproducible results, which is something valuable. But now I come to the most important slide, which is the feature concept of CPP. So a feature here is defined as a combination of a part is split into scale. A part can be the TMD or the JMDs. Now we consider the TMDs of APP, which has 23 amino acids, and the one of notch 1, which has 21 amino acids. So we cannot compare it position-wise because they have a different length. So to make these TMDs comparable, we can split them in segments. For example, here we split them in 5 segments and consider the first one, giving us 5 amino acids for APP and 4 for notch 1. As well, we can consider patterns because the helixes you have seen before, every third and fourth amino acid, the residues are on the same helical site, so they can form an interaction with other proteins, which is essential. And now we can take a scale, for example volume, assign the scale values to each amino acid, compute the average, which results in the feature values. CPP is computing such feature values for all proteins in the reference data set and in the test data set, and looking for the features which have the strongest discrimination between these two data groups. And here you can see the top 15 features. So we have the scale subcategory and the positions highlighted in gray. And now we have the mean difference of the feature values, where red indicates higher values for substrates, blue indicates lower values for substrates. We use machine learning models to rank these features, and what you can appreciate here is that the most important feature with 5% importance is a positive increase or an increased beta strand tendency in this area, which I cannot show, but you see this gray, it's the gray part on the top, and this is actually where the cleavage is taking place. Okay, we can now map to get a single residue explanation or information which is important to perhaps understand mutations. We can map the importance on the residue position, and here 2 sequence regions are striking out: the cleavage region with 33% and the other region with which we call the TMDC anchor with 28%. And this is shown in the CPP profile. And we can now add another layer of complexity by integrating the scales. And this is shown by this CPP feature map, which is actually revealing the common yet unknown property as what we call the physicochemical signature of γ-Secretase substrates. So what you have here is, again, the mean difference between substrates and the reference data set. Red indicates higher values for substrates, blue lower values for substrates. And we have the feature importance indicated by the black squares, both given per scale subcategory and residue combination. And this is a very complex map, so our collaborators were studying this for days. And to simplify this a little bit, let's focus on this part. We call this a well-defined TMDC anchor, characterized by an increased accessible surface area, side chain length, helix termination, and so forth. And now something puzzling appears. Another important group of features is this conformational preference around the cleavage site for a helical and a beta strand extended tendency, which actually doesn't make sense in the beginning because we do know they are helixes, they are not extended. This was really puzzling in the beginning. However, shortly after this initial results, this publication came out. So this is showing us if it's unbound, APP, the substrates, they're helical, but if they interact with γ-Secretase and if they're bound, the helical conformation can unfold and form a hybrid beta sheet together with the enzyme, with γ-Secretase. And in this transition between helix and extended conformation, cleavage can take place. So this was a really big endeavor, big breakthrough to release this structure. And actually with CPP, we can see that this property is actually a common property of all substrates, not only of APP. This is now the extended conformation I promised you, and this shows that CPP can explain how context-dependent conformational changes can be used to, for example, for machine learning predictions. Okay, we benchmarked, of course, we had such good features. We benchmarked them against state-of-the-art methods. There we used protein language models-based protein embeddings, so namely Prot5 from the Rost lab. And you can see in blue that actually they only achieve 2%, only 65% performance, while our approach achieved 90% prediction performance. And we went to the lab and tested some of the predictions with overexpression cleavage assays. And here you see that in the presence of the inhibitor, that you expect an accumulation of the so-called CTF, which validates predicted substrates as substrates, which you could do for 11 substrates in total. And for the non-substrates, you expect no change, which you can see here for 5 non-substrates, which yields a success rate of in total 88%, consistent with our computational results. So lastly, I wanted to combine the CPP feature concept with SHAP. So we heard about SHAP. Instead of feature importance, you get feature impact, which can be positive, indicated in red, or negative, indicated in blue. We can see the high prediction score of APP can be explained by the positive impact of all its features. We can now map this to the sequence of APP. And there you can see that the TMDC anchor and the cleavage site shows the strongest positive impact. Now we can look at the less good substrate, BTC, 67% prediction score, can be explained by the negative impact of the residues within the TMDC anchor. Now we look at the very good non-substrate, TMX3, and the low prediction score can be explained by the overall negative impact of the features. Okay, with this I come to an end. To solve the long-standing problem how gamma-secretase recognizes the substrates, we developed different methods, including CPP most importantly, and this helps us to identify this physicochemical signature. And we could outperform Prot-5 embedding, so protein large language model-based approaches, which is consistent with our experimental validation. And by combining CPP with SHAP, we reach single residue resolution explainability. And I hope that CPP can help us to open new venues to understand how sequences are actually encoding molecular recognition. And the tools I showed here are accessible as a Python package, AA analysis. And with this, I come to an end. Thank you to your attention. Of course, thank you to the Steiner lab, Harald Steiner, Fritz Kamp, Dimitri Frishman, and the other collaborators. And thank you very much for your attention. And I'm happy to take any question.
Any questions? Come to the microphones. So I'm wondering here, you indicated also in the end, well, the framework is available. Are there cases for other proteins that you already know of? How does this transfer to other? Well, I'm not talking here about cross-domain transfer. Maybe that's also a question. Or I don't know how this could transfer to other sequences outside the biomedical domain, but within the biomedical domain, have you other cases? Is this upcoming?
So we are applying this on other intermembrane proteases, or for other proteases, which is an obvious extension. And for signal peptidases, for example, we try to use this framework as well to design proteins. So since this was as well outlined in the keynote, that we have this vast search space, and if we know which sequences, which residue is most important, we can minimize the search space or guide it more. So we try to develop this further for protein engineering as well. So yeah, there are many things I'm working on with my students, and I hope that soon you will see more of our work, of course. Thank you for the question.
One question.
I think I just realized that you answered the
And it was like, okay, this has to be true because that's the true underlying signal. And then they were convinced, okay, we invest a lot of money to do the validations in the lab. And this is, I think, the key thing. So explainability builds a bridge between us, between the computational scientists and the wet lab people or the other application-focused people. And this is important. And we get an understanding. So these are the two key things about explainability in my example, but I think I can speak as well for you, that you have similar.
So effectively, you're using this for using a model where people know what the ground truth is. You show the model learned the correct ground truth, and then you extrapolate to new scenarios, and they are more likely to trust this.
Yes, and in long term, I want to combine this with causal modeling, and then we get a better understanding and we can do a consistency check, for example. Mm-hmm. But yeah, that's at the beginning of this exciting field.
All right. Then let's thank the speaker again.